Hello, my name's Tom McLaughlin. I'm in the School of STEM Education at Dublin University. I'm also the curator of the DCU Science Archive and Herbarium. Starting with this paper, we're looking at material culture and science education as a resource for today's science. So rather than keeping old stacks of equipment in schools and not using them, I want to advocate for the use of historic equipment in schools, and that I want to argue that many of the reasons that we think we've stopped using things aren't particularly valid, and that we should re-engage with whatever equipment's in school and work with that. Teachers have often reported that there are a number of problems with science, such as lack of resources and money to purchase new equipment, um, lack of time to prepare practical work, lack of expertise in the subject, though that may not necessarily be admitted by teachers, and lack of craft skills for repair and maintenance of equipment, which is not necessarily a problem that we can address. But certainly in the first few we can. So issues relating to resources, if we think of them in a Venn diagram, they would intersect with competency and craft skills. Similarly, that would issues in time and the time to do certain things and so on, that would intersect with both of those. But where does expertise lie? Expertise, with greater expertise, time management is improved. With greater expertise, management of resources is improved. And with greater expertise, obviously you would have more craft skills. So it seems that expertise is the one that is the origin, or the one that connects all of these, and perhaps it's the three-way intersection is where expertise lies. So I'm going to start with three simple examples of historic biology experiments of material culture and how we still can continue to use these. And the first example is a respirometer. And the author was asked recently what this apparatus is and what it is for. Now, it has two functions: determining the rate of respiration and calculating respiratory quotient, which is a mathematical version of the first one. And the apparatus, though, does appear to have a fairly high demand in terms of pedagogical content knowledge, PCK. So it's fairly simple to use once we are familiar with it. But the fact of the matter is many teachers, when they're shown a picture of this, they have never seen one of these pieces of equipment, or they don't know how to set it up and use it. Okay, so it's so that would be a curious thing. The second piece of equipment is a potometer. Again, I asked a class of prospective biology teachers, is this something that they would have seen? And they said no. However, transpiration, and it's a classic experiment in school biology, and the key thing to remember is that when you're setting it up, that the plant material needs to be woody shoots, and it needs to be all assembled underwater. There is one aspect that a small amount of water is expended in photosynthesis, and that's not taken into account as the air bubble moves up. Once broken, though, few teachers are able to fix it. And then the third thing is the leaf clip, sprung leaf clip. It requires expertise in the handling of pot plants, knowledge of purging the leaf starch, and testing the leaf for starch. And although it's a simple apparatus, the PCK demand is quite high because there are three contingent concepts. And again, teachers wouldn't recognise the piece of equipment, but they would soon work out, because what we tend to do in Ireland is use tin foil pressed between two slides instead of this actual piece of equipment. What do they all have in common? Well, they were all invented by William Francis Ganong in the late 19th century, and their use was a staple of botany and then biology teaching throughout the 20th century, and they could be replaced with everyday items or general lab equipment. The thing is that Ganong didn't so much invent them as he took the ideas that he was trying to teach, and he developed pieces of equipment or adapted other pieces of equipment that would help in the teaching of these concepts. So they are properly pedagogical tools. In Ireland, they would have been used to teach concepts relating to respiration, transpiration, and photosynthesis throughout the high school, which still remain on the syllabi. So, if they're still on the syllabi, why are these equipment not being used? That is the question. And partly it falls that teachers feel they haven't the time to run these experiments. They feel that they haven't encountered them before, so they don't have the expertise. The thing is that they're generally certainly the first two, such as the respirometer and the potometer, are actually on the official government lists, so they should have been supplied to schools, but teachers aren't recognising them when they actually see them in the prep room. So there's definitely scope for getting teachers to re-engage with equipment. I'm going to go to a more complex example now, and that would be the Shandon chromatography and electrophoresis kit. It was developed in the 1960s, manufactured sometime after 1970. It's a large 2.5 litre glass jar with blue plastic lids, clips, and tubular electrodes and solvent dish. It was supplied to Irish schools as item SSCB 28 official government science resource list. Okay, so what we're going to, you can see here, there's three ways it could be used. In this top left here, that was ascending chromatography. In the diagram on the right, that is descending chromatography, and the third one at the bottom, number 3, that is in electrophoresis. Paper electrophoresis, a very simple form of electrophoresis, but still covers all the concepts. So what we're going to look at is that where is teachers' knowledge of electrophoresis, and where does that stand, and would they be open to going with that? So carried out an interview, 10 teachers with more than 10 years practice. The purpose was to establish a baseline. The questions: have you ever taught content employing electrophoresis? Have you ever conducted a practical lab session with electrophoresis? Were you aware that it was in the government list even after the item was no longer manufactured? Do you remember seeing the Shandon kit in schools yourself? Did you use it? Did you know it was suggested as a mandatory activity in the 1994 spec? And how would you feel if we were encouraged again to teach it? And you can see that the questions are essentially dichotomous, but the teacher is encouraged to expand their ideas and provide anecdotes. So what we see is that the results actually provide scanned information on the use of electrophoresis in the sample of teachers. Some of the cohort had not heard of the technique, while others thought it was used exclusively for DNA separation as opposed to any charged species. There was no sense that the teachers had actually heard of RNA Theselius and that he was building on earlier work from the beginning of the 19th century. And those who had heard or mentioned electrophoresis considered it only be done using agarose gel, no other medium. It was further perceived as difficult and expensive. Teachers were generally unaware of the listing SSCB or to include electrophoresis, nor remember any inclusion in the 1994 draft syllabus, and the general consensus was teachers should be trained in what they should teach. Okay, and I think that's very important, because people are doing degrees in marine biology, they do degrees in botany, they do degrees in biotechnology, and so on, and they all become biology teachers when their experiences are vastly different. And it should be the case that whatever intervening training that should take place, they all should be trained in what takes place in schools. That's what they should be actually trained in. When something new comes in, it should be training. It's notable that when there is a new syllabus, that any training is just on the, if you like, the administration rather than the actual practice of teaching. But teachers did respond positively to the idea that their predecessors stopped the inclusion of electrophoresis in the 1990s. The questionnaire results then, it's split really. There's a HPS, that's History and Philosophy of Science question, about the date the principle was discovered. If they knew the decade in the 20th century, Nobel Prize was awarded to RNA Theselius. And then the technical concept on the ions, the medium, and the broad use. So if we look at this, we see that you get an almost quartering of the pie chart when it's which is a guess, and there's no strong answer, and it's early 19th century. It's the correct answer. The electrophoresis depends on existence, and it is Zwitter ions, which is 36.9%. But you can see there's things called quaternions, which are mathematical concepts. Is the next one. They didn't really know which what we were talking about there. It was 1948 was Arne Tiselius's thing, Nobel Prize, and they're very much focused on understanding that electrophoresis uses agarose gel rather than other media. And in my own training, I would have done membrane, cellulose membrane, which is very similar to paper but more technical. And gel electrophoresis was just really coming in at that stage. And then we have electrophoresis. The general answer correctly is the separation technique of charged compounds, which is correct. So basically, we can see that that even though that DNA science is part of the upper high school in Ireland, that there's very poor knowledge of the technique of analysing it, that is by electrophoresis. There is equipment that can be used to help teach the concepts. You know, if you aren't actually doing the gel electrophoresis, you certainly can do some form of electrophoresis in order to teach the concepts, you know. And since biology is an experimental science, you know, the practice of that is important, if not critical. Okay. Experimental science, biology is actually part of the learning with objects paradigm in education, that we see, whether the objects are organisms, the chemical constituents, or even whole ecosystems, and the tools, apparatus, and equipment to study with, and all can come into play in order to teach biology. And there's very little that is redundant. As we can see, both teachers and science students have issues with electrophoresis. Their knowledge is scant and their practical use even less. And just a note then that in the Science Archive in Dublin City University, we have Shandon Unikit, and we teach the practice of electrophoresis papers, the medium, discussing the theory of the technique and Tiselius's contribution to science. And all these gathered together as tools for learning in the chat triangular framework of Engelstrom. Okay. So thank you very much for your attention.